Hello everyone. Myself Swati Tiwari and I am a student of Jawaharlal Nehru Krishi Vishwavidyalaya College of Agriculture Rewa. Today I am presenting my topic on hybrid seed production of rice under the guidance of Dr. R.P. Joshi sir. So let us first talk about the botanical classification of rice. Scientific name Oryza sativa, family Poaceae, chromosome number 24, origin Southeast Asia. Hybrid seed production in rice. First, earlier farmers used to cultivate improved rice varieties, but due to open border, they came contact with Indian and some imported Chinese hybrid rice varieties and given interest in hybrid rice farming. Second, hybrid rice give production of 20 to 30% higher than inbred rice varieties, which can make small farmers and individual families to boost crop yield dramatically. Third, Nepal Agriculture Research Council started to evaluate Indian and Chinese hybrid varieties in collaboration with concerned seed companies to get registered the varieties. Three line system. This system, three line, first, cytoplasmic genetic male sterile line, also called A line or female line, maintainer line B, and restorer line R are required. CMS lines should be expensively evaluated for their pollen sterility and panicle exertion along with the traits. Pollen sterility test along with some evaluated CMS liners. Development and evaluation of CMS line. Development of maintainer line B. Identification development of restorer lines R. Production and evaluation of F1 hybrid seeds. Maintenance of ABR line. Multiplication of parental line. The seed of male sterile line is multiplied by crossing A and B lines in isolated plot. There is a small difference between the growth duration of A and B lines. Their sowing dates are adjusted to achieve good synchronization of their flowering. The seed of parental line should be genetically as well as physically pure. B and R lines are also multiplied simultaneously in separated isolated plots. This process is undertaken by breeder. For large seed production, breeders and specialized institute, companies and group could also be involved. Hybrid rice seed is produced to grow hybrid rice by the farmers. Here are some hybrid varieties of rice like Jaya, Ratna, Sona, Swarna, PRH10. Tree line system, hybrid seed production in rice. Seeding and transplanting. Sprouting seed should be sown uniform ally in a well-leveled wet seedbed at the rate of 40 gram per meter square as on wet seed basis. For example, for 1 big hectare transplanting, 4 katha land is required for seedbed. Level of seedbed plot is necessary, otherwise, seedling will be damaged in lower parts of the seed bed by waterlogging and uneven growth in upper parts. Transplanting could be done when plant gain enough height to uproot the seedlings. Transplanting could be done easily when height of seedling is 15 cm or when seedlings are of 5.5 leaf stage. Transplanting of older seedling delays flowering and transplanting of younger seedling advances flowering. Some transplanting works are shown in photos 12 to 15. Transplantations should be done 1 day later after field preparation. If seed parent is 10 days shorter earlier in growth duration and pollen parent seed R line is splitted into two seeding dates as R1 and R2 lines for raising of seedling as they can also be named as R1 and R2. In short, R1 of pollen parent could be transplanted first in paired rows followed by A lines in a recommended row ratio when seedlings get ready. If seed parent is greater than 10-day longer growth duration than pollen parent, A line should be transplanted first in recommended ratio, keeping vacant space to transplant R2 lines in a pair rows later. Rogging. Person involved in rogging should be aware of characteristics of varieties to be rogged. Rogging plays a vital role for maintaining seed quality and purity. Rogging is the removal of undesirable rice plants and the weeds from hybrid seed production plots. Unwanted rice plants are those plants that differ from true to type A or R plants. Isolation. Isolation is necessary in seed production plots to check outer pollens from adjoining fields for contamination. Pollen in rice plant is small in size and can move longer distance with the wind. So isolation could be provided by the following three different ways. In small amount for initial research on hybridized crossing chamber can be built. Harvesting and seed yielding. Harvesting. The crop should be harvested when the grains are hard and yellow with the moisture percentage of 23 to 24%. For combined harvesting, the moisture percentage should be in the range of 16 to 18 centimeter. The crop is cut at the base with the sickle and the plants are left in the field for 2 to 3 days. Then they are shred on clean threshing floor or tarpaulin. After winnowing and cleaning, the seeds should be dried to save moisture limits of 13% before storage. Seed yield. The seed yields are in the range of 5.0 to 6.0 ton per hectare, depending upon the variety and the management practices adopted. Thank you.